Hello everyone. Today I will start with the topic cell division. Cell division is the biological process by which a parent cell divides into two daughter cells. Parent cell is the one which will undergo division and daughter cell is the one which will get formed after undergoing division. Now what are the reasons for cell division? First is your growth, repair, replacement and reproduction. We know that every organism starts their life from a single cell. This cell is going to immediately divide and form a cluster of cell to form the tissue and organ. Hence, it is going to be essential for growth. Next, it is required for repair and replacement. Any kind of wear and tear of the tissues in the body or consider any accidental injury that may happen during our lifetime will require repair of such injury, for which cell division is important in order to join the broken ends. Finally, reproduction will also require the activity of the dividing cell. However, the type of cell division in these particular three conditions will be comparatively different than that of reproduction. There are two types of cell division. One is mitosis and the other is meiosis. Cell division leading to growth and development is known as mitosis. Okay, and cell division leading to production of gametes, which is also known as the sex cells, is known as meiosis. Okay, so in this particular type of division, it is going to lead to reproduction only, while mitosis will be leading to growth, repair and replacement. Mitosis is the type of cell division in which one parent cell divides into two identical daughter cells. One of the most important part is that the chromosome number is going to be maintained in each cell division. See, in our human cell, there are 46 chromosomes, which can also be written as 23 pairs. Okay. So this point means that the number of chromosome in parents will be equal to the number of chromosome in daughter cell while undergoing cell division through mitosis. And the last point states that there are 4 phases in which mitosis will take place. First is your prophase, then it is your metaphase, anaphase and telophase. Now we'll be studying these 4 in brief. Before we start with the actual cell division, there is a phase which is known as interphase, also known as resting phase. During this period, no visible change in chromosome is observed. However, there is active synthesis of DNA. The first diagram is going to represent our animal cell. Okay. And the second one represents our plant cell. See, in the animal cell, we have got the centrosome with the centrioles present in it. We have the chromatin threads inside the nucleus and also nucleolus is present inside it. Same things will be there in our plant cell. Only missing things will be the centrioles. There will be no centrioles. While what is present will be our cell wall. Cell wall is present. The first phase of our cell division is prophase. During this phase, the chromosome becomes short and thick and will be clearly visible inside the nucleus. Okay, these are our chromosomes and they get duplicated to form chromatids. chromatid. The point of attachment of the chromatid is known as centromere. Okay. Now see, the centriole is also going to split into two and will move towards the opposite end at the later part of prophase. Okay. So late prophase, they will move towards the opposite end. Also, a number of fiber called spindle fiber will appear between the centriole, spindle fiber. Okay. On the other hand, in plant cell, those spindle fiber will appear, but there will be no centriole at the opposite end. Also, if you see, nuclear membrane and nucleolus will disappear. So, these are some points which need to be kept in mind. Centriole move apart and reach the opposite end. This is our first point. Next, the chromosome is going to become distinct and duplicated as chromatid. Sister chromatid attached at centromere, spindle fiber appear, and nuclear membrane and nucleolus will disappear. The second phase of cell division is metaphase. During this phase, the duplicated chromosome are going to arrange themselves in the equatorial plane. This is the equatorial plane. Okay. Also, here each chromosome get attached to the spindle fiber by its centromere. So, in this way, the chromosome get lined up in one plane at the equator. The third phase of cell division is your anaphase. During this phase, the centromere attaching the two chromatids will divide. Okay. Each chromatid basically gets pulled towards the opposite pole, and this action will be carried out by the contraction of the spindle fiber. So, this contraction is going to help the chromatids reach the opposite poles. This phenomena remains the same in case of plant and animal cell, with the centrioles missing in case of plant cell. The last phase of mitosis is your telophase. Okay. In this phase, each chromatid or the daughter chromosome will lengthen, they become thinner and will turn into chromatin threads. So, these are our new chromatin threads in the daughter cell. Okay. The nuclear membrane has split it into two, you can see, with the nucleolus present in each. Nucleolus. The nuclear membrane is basically formed by furrowing in case of animal cell. So, the furrow, this is our furrow, which is basically dividing the cell into two, while in case of plant cell, it is the cell plate or cell plate will get formed by this cell plate, which grow from the center to the periphery to form two cells. So, this is our parent cell which divides into two daughter cell. Also, spindle fiber will disappear now, nuclear membrane will get formed and nucleoli will again reappear. This is how cell division occurs. The second type of cell division is your meiosis, okay, which will take place only in the reproductive organs. In this type, the number of chromosome in the sex cell will be halved, okay. So, basically 2n is your 46 chromosome and n 46 chromosome and n will be your 23 chromosome, and this is known as haploid condition. 2n is your diploid condition, okay. So, here only half the number of chromosome will be passed into the daughter cell. Next, the paternal and maternal chromosome will be mixed up. So, basically during the separation from the homologous pair, the mixing will take place. Finally, is the cross joining or exchange of chromatid material will take place. While the paternal and maternal chromosome are separating, the chromatid material gets very often exchanged, and therefore in the offspring, both the characteristic of the parents will get observed. So, these are some significance of meiosis. Anything more in the syllabus is not included. So, this much will do. Based on some points, we can differentiate between mitosis and meiosis. Where it occurs. Mitosis will occur in the somatic cell. The term soma means body, and meiosis occur in the reproductive cells. What is it required for? It is required for growth and replacement, and meiosis for gamete formation. Daughter cells produced in mitosis, 2 daughter cells. In meiosis, 4 daughter cells, because each parent will give 2 daughter cells. So, 2 plus 2 leads to 4 daughter cells. Number of chromosome passed, Full set of chromosome means the diploid one, or double the number of N will be passed. And in meiosis, half the number of chromosome, because half of 2N is N, so it is known as haploid. Identity of chromosome, it will be identical, means exactly the same. While meiosis will lead to random assortment between the gametes. This is what leads to genetic variation, and the offspring will look somewhat different or different from their parents. Okay. That is it for this video. In the next video, we'll discuss about cytokinesis, karyokinesis, some important information about the cell cycle, and the important questions which are very likely to come. Thank you.